I am Mohammed Bentires Alj. I'm a professor at the University of Basel. The goal of our research is to understand breast biology and breast cancer and to use this knowledge to improve therapy for our patients in Basel and throughout the world. My lab focuses on the cell autonomous and non-cell autonomous mechanisms regulating normal and neoplastic breast stem cells, resistance to therapy, and metastasis. In collaboration with clinicians from the University Hospital of Basel, we are developing a breast cancer personalized medicine program. I'm pleased to welcome you to conversations with key figures in mammary gland biology and breast cancer.
All right, I am Mina Bissell. I work at Lawrence Berkeley National Laboratory. My title is a distinguished scientist. It's not actually an appendix, it's an actual title. So I'm old enough to have deserved it. And I work on breast cancer and breast development and the question of organ specificity.
Mina, we first met at the Mammary Gland Biology Gordon Conference. You must have attended many of them. Can you tell us about this must-go meeting of our field?
Well, you say this must go. By the time you get to be my age, there are only so many places you can go. So unfortunately,
so you are not coming to my meeting.
Well, you remember, I am sending somebody very important for you, right? Is
Absolutely.
He coming? Yes. All right. So, this is one of the earliest Gordon conferences I went to. I must say, I'm just trying to remember, was it a mammary gland Gordon conference, or was it an adhesion conference, or was it an extracellular matrix conference? You know, because my work sort of has come more and more to be mammary gland, and it really didn't start with mammary glands. So in the early days, I would attend the Gordon conferences in cancer, Gordon conferences in extracellular matrix, and then when I began to want to prove the principles that I was very excited about, then what I did was that I started going to the mammary Gordon conferences. And it really was enjoyable. And it was 1 year in sort of basic biology of the mammary gland, and another year on milk production. Now, my very first postdoc who worked with me on mammary gland biology was Joanne Emmerman. And Joanne Emmerman had worked with Dorothy Pitelka, and I wish you could have interviewed Dorothy Pitelka, and you still can interview
Absolutely.
Joanne Emmerman because the two of them had done something important. And when she came to do postdoc with me, I decided that the best way to try and actually answer the question I was interested in would be to use mammary gland, because it's such an amazing organ. It goes from virgin to pregnancy to lactation to involution, and you take the babies away and it comes back and does this. How does it do it? So that really got me excited. So I have attended many of them, and I think I even ran once to be the chair, and I have given the keynote or whatever for two of them.
Okay, yeah.
But I didn't get elected when I ran for chair, and we were all laughing because we were kind of laughing about doing a political system, you know, like Democrats and Republicans. Republicans won. Okay.
So when I went through your CV and achievements, I was impressed, but not surprised, by the number of awards and honors you obtained, including your election at the National Academy of Science of the United States. This is a great honor. What does being a National Academy member involve?
Well, I never know. The election to National Academy is a very involved process. And I must say, I don't think we have people there who don't deserve it, but we have a lot of people who deserve it and are not there.
Okay.
So the question is, what are the elements that go into eventually getting elected? I think what it takes is few of your colleagues and peers who already are members of academy to really push for you. And believe me, it is very complex, because in the section that I got elected to, which is cell biology and developmental biology, which is a very prestigious one to get into, at most we can elect 3 people a year.
Okay, only.
And there are other, of course, sections, in genetics or in medicine or whatever, but they can't do, some of them can do only 1, and others can do only 2, but hardly anyway. So at the end of the day, you have something like 90 people in your list, and at the end of the year, you end up with 2 or 3, at the most, usually 2. So it's a very involved process. I'm very honored to be a member, and I know colleagues pushed for me for years and years and years. And with some people, it doesn't take that long. With some people, it takes longer. And as you know, I was a rebel. And half the people in the academy, in fact, most of the biologists in the academy had no idea why I was doing what I was doing. And also, I'm very outspoken. And sometimes that scares some people. Mm-hmm. And it's not a very, they just don't, they have to really get to know you. But luckily, I ended up having a couple of colleagues who were persistent. And towards the end, some other people came in who were guys who had heard me give a talk and were impressed. And then they started reading my papers, so they pushed. And I'm very honored to be a member, but honestly, if you want to be a true member, and not just wear it like a badge, it's a lot of work.
Yeah, it requires time.
And even though I can't go every time, but I just reviewed, for example, a very important report for them. These things takes time, they have committees, and I'm organizing another meeting between the institute, because I'm also the Institute of Medicine member. And there are 3 academies. And this is called the Institute of Medicine. It's another national academy component. And the third one is Academy of Engineers. And the 3 of them are called national academies. And so I'm also a member of IOM. So between the IOM and National Research Council, who have people from each academy, and they do all their sort of planning in certain things. They have now gotten involved with Keck Foundation, and they have a very interesting meeting that takes 2 years to organize. And now I have gotten involved in one of those.
The organization of the
for Keck Foundation to ask some really fundamental questions.
That's exciting.
It's going to be fun. This one basically asks the question: how do individuals behave differently from groups? And it doesn't have only to do with people. It has to do with bees, it has to do with plants, and it has to do, in my case, it has to do with one cell as opposed to a community of cells which make an organ.
Yeah, it sounds like an exciting meeting.
Yeah.
So Mina, you studied chemistry at Bryn Mawr College. Mm-hmm. Then you obtained your bachelor at Radcliffe, Harvard Medical School or Harvard College, and a master in bacteriology and biochemistry at Harvard University Medical School. Then you obtained a PhD in microbiology and molecular genetics at Harvard Medical School in 1969. Can you tell us about your first years in research?
Well, I come from Iran, and I came here when I was barely 18 by myself to go to Bryn Mawr. It was quite interesting, and that's, I think, is one of the best schools in U.S. Honestly, having gone to Radcliffe, which then the year I went to Harvard, I in fact got a Harvard degree, and I was not happy. I wanted it to be in Latin, and it was in English. But I went to Bryn Mawr at the suggestion of an American who was the head of American Friends of Middle East. And if the truth has to be known, later I found out they were a branch of CIA. And the head of the branch called my father, and he had seen my application, because I didn't know where to go. No, nobody, my family had gone that young to do something. But I had a very special scholarship. I was the toppest student in the country, and I had a whole lot of other little things added. And so the guy invited my father and said, your daughter should not go to University of Pennsylvania or Michigan or wherever it is she's applying for. She should go to Bryn Mawr. And if I had never heard of it, and it is one of the 7 sister colleges. Okay. The way you have 7 Ivy League for the men. And it was for University of Pennsylvania, and for Haverford was not quite Ivy League, but they, So to make a long story short, I went to Bryn Mawr. It was the best experience of my school years, ever. And I'm sad to say that it was only 2 years, because I transferred as a chemistry student to Harvard to get married. So, and I think Bryn Mawr is better than Radcliffe. I think it's better than Harvard. It's better than Berkeley. It's better. Have daughters, send them to Bryn Mawr. It's an amazing school. And I was fortunate.
And it's still good today.
It's fantastic. And they're the only ones I give a lot of money to. Because they bring a lot of foreign students. They do a magnificent, it's a 1-to-1, and they have remained a girls' school, even though they have guys from Haverford and from University of Pennsylvania, from other places, staying in dormitories and taking classes, but this is essentially still a women's school. And it's a magnificent school. It's beautiful, it's Gothic, it has all this lovely ground, and they're doing magnificently. Anyway, I had no idea what I wanted to do. So I debated between chemistry and English literature. I had a wonderful English teacher who is just 90 or a little bit more. She's moved to Boston to be a professor later on, Anne Berthoff, and she was so fantastic. And I am still a friend with her, and I loved English literature. I always have loved literature. I still do. I read a lot, and I love poetry and And then I finally decided, well, English I can maybe do somewhat on my own, which is actually not true, but never mind. And then I went toward chemistry, and it was easy for me. And it had something to do with our education back in Iran, where physics, chemistry, math, these things, we had a lot of it, and I was good at it. So it was easier, actually, in some certain sense. But I went towards chemistry, and when I went to Radcliffe, I won the first year. It was still called Radcliffe. Then in the senior year, even though they were taking classes for years together, they didn't give Harvard degree. They weren't calling it Harvard, and I won the Medal of Institute of Chemists, and then for various reasons, I ended up at Harvard Medical School, and the rest is history. We'll talk about.
So that was where you did your PhD and started research?
I did my PhD at Harvard Medical School. I also got my master's there. They give you a master if you're a PhD student. After the first year you do well, they give you a master's.
So what did you work on for your PhD?
I was a bacterial geneticist, and my professor was a guy called Luigi Guarini that not very many people know, but he passed away a long time ago. He was an older guy. I think he was about 70 when he tapped me to be his graduate student. I was taking a class, and they were showing something about cameras and EM, and it had some physics in it. And in those days, I could still do it. So he was there, and then he sent somebody from his lab to say, tell Mina to come talk to me. So I went to work with him, and I did a thesis bacterial genetics, but really it was bacterial physiology, because it had to do with secretion of a bacterial proteinase. And the mechanism of how it was regulated was completely unknown. And if you read a one-page thing I wrote for Nature Cell Biology 3-4 years ago, which called Turning Point, and they ask what in your life happened that turned your work, I wrote a little bit about my professor and about my interaction with him and about the work that I did and how I completely disagreed with him after 3 or 4 years about what this phenomenon he had observed, and nobody else in his group was working on that. And I took it because I had a baby. I just had a baby first year graduate school. And those days, nobody did this. So, you know, we had 3 women in the medical school class and 200 men, and 1 faculty.
So where was it? Is it now where the quad is now?
Yes, yes, exactly.
Okay.
Yeah, exactly. Yeah, that's where a lot of,
That's where cell biology is now.
Yeah, the cell biology is now, microbiology is now, the genetics is, you know, they're all.
Absolutely, yes.
In those days, they were fewer, all these other hospitals were not so many. Anyway, so that's how it happened.
So Mina, before talking about your scientific career as an independent investigator, can you tell us a bit about your personal history, childhood, birthplace?
I already preempted you.
Yes.
So, of course, I come from Iran. I came from a very educated family, and my father's family were absolutely the most amazing people. My aunts were professors, either of literature, or my aunts were doctors, and my other aunt on the mother's side was the head of immunology at Tehran Medical School. And most women of my kind of family, which was upper middle class, Iran was and it still is a class society. America has become the worst class society. But it was sort of more like France and England, so it had layers. And so people were all educated. And education and achievement in the history of this 4,000-year-old country is really one of the most important. It wasn't the money per se. And in fact, upper middle class people were maybe coming from backgrounds of money or whatever, were not necessarily rich by any means, but they were still up there, and they had servants, and so women could do what they could do. They had grandmothers, and they had servants, and they had nannies, and it brings me back to the idea that was so unknown in U.S. when I first came. Hardly any woman was being encouraged. I mean, women who went to graduate school and did whatever, did it either because they were immigrant families or because they did it against the wishes of, So for me, this was, if I didn't get a PhD, my parents would have been very disappointed. My father had a PhD in law. He was educated in France. My mom was very unhappy because she didn't finish college, because my father wouldn't let her. And so she, he had said it's difficult. And so we were 2 daughters, and I was the younger, and I was raised just like a boy, And in Iran, women do very well. Of my 40, 50, second, third, first cousins, the women are all over the world doing magnificently.
Fabulous.
And even now in Tehran University, I haven't been back since the revolution, partly because I'm so outspoken, but even now I am told that entry class across the country in engineering, architecture, and medicine, 58% are women. And 52% of the faculty in Tehran University are women. Can you imagine? I mean, people's idea of Iran is like what Afghani women wore, the burqa or whatever. you know, if you look at the rebels, they do, you know, but it's the culture, it's the, you know, we used to have a queen, and it's the context, and it made me who I am. And a lot of people in US always say, aren't you happy you ran away? I said, I didn't run away. I came to be educated, and then I got married, and, you know, but I do what I do precisely because I come from Iran.
So what made you go to science and not philosophy, or?
Well, I talked about this, partly because I was always good at it, you know, and also I was very curious. I was very curious. And I made very funny choices because I had nobody in my family was a scientist. They were doctors. And my aunt had done some research in immunology, but she was not my role model, not at all. I only read a book of Madame Curie probably when I was young, and people were laughing about the fact that I used to do funny kind of experiments, like there were a lot of hype and I'm trying to think of the word, you know, when you believe in all these things, that, for example, that if you have a frog, you get a little thing in your hand, if a frog touches you, you know, superstition.
Yeah.
And I was always, ever since I was little, I was fighting against it. So I remember I had a nurse, and they believed that if you pour hot water from a balcony down, you would get these lesions in your hand. And it's a good thing to believe because you shouldn't be putting hot water. But I said to her one night when my parents weren't home, okay, you're going to sit there, I was 4, and I'm going to pour the hot water.
Experimentally.
So, you know, I like to find things, and I like to argue things. And my father was a lawyer, and he would debate us across the table, and, you know, and I was on a stage ever since I was young, so that's how I give talks, you know. So I think that's just naturally pushed me toward.
So where did your interest in cancer originated from?
I have no idea. It just evolved. The same way that my chemistry evolved and the bacteriology evolved. My reason for doing bacteriology was because I just thought, well, I can't be a medical doctor because I want to have a family and I want to have children, you know, just thinking completely the wrong way. And so I went to bacteriology and I liked it. It was a challenge. And then when I decided to come to California is because my husband, who was a medical doctor at Harvard Medical School, got a job back in California where he was born. And so we were coming with my daughter and my husband to California. So he was at UCSF, and I had to think of something to do, and I read a paper of, or some report in New York Times which said that a professor at Berkeley had found some protease. And since my in virally transformed cells, or, you know, using Rous sarcoma virus, and since I had done my PhD thesis on protease, then I applied to a number of fellowships. I got them all, and again, I was very naive. I chose the American Cancer Society, and I'm glad I did in retrospect, but I got that fellowship. So I went to Berkeley to do a postdoc, and
So this is what, you know, this is what, you were Milton Fellow, or Milton?
No, the Milton Fellow at Harvard had to do with aspect of my thesis where I was proposing the first model of co-translational secretion. And this is one of those models that people later got very famous, but I was saying that molecules get out from the membrane, that they're synthesized, you know, bacteria have a wall. And so between the wall and the membrane, there is this space, and I was saying that the ribosomes had to be membrane bound. Remember, this is 1960s. It is still, people didn't know that. And in order to try to make sense out of my PhD thesis, I came up to the idea that the proteins get out unfolded, or at least this protein. But it would get out unfolded, and then as soon as it would come out, it would meet calcium and then gets folded, while my professor idea was something completely different. And so it was a proteinase that I was working with that was doing this. And my professor at Berkeley was a guy called Harry Rubin, who was actually a veterinarian.
By training,
yeah, and he wouldn't use radioactivity in those days, and so it was quite a different experience for me to come and do that. But he had said that these proteinase is secreted actively to do something, and I showed that in fact that wasn't the case, and that was my first paper from his lab, where I showed that, and the way I did it is that I took something like lactic dehydrogenase that is supposed to be intracellular, and I showed that anytime we had proteolytic activity, LDH was in the medium, so it was due to lysis, and we published it.
Very nice, very nice. So in 1972, you became staff biochemist at the Lawrence Berkeley National Laboratories, the LBNL, and senior staff in 1976 in Berkeley, and now you are, as you mentioned, a distinguished scientist at LBNL. Can you tell us more about LBNL?
Sure. So very few people understand the exact location and the meaning of a national laboratory in the US. So LBNL is right out above Berkeley. And the guy who founded it was this guy Lawrence, who did the first accelerator, and he won the Nobel Prize for it. And then later, of course, it got bigger and bigger and bigger, and went to Livermore, and people were doing defense-related stuff. And then he went to Los Alamos, and the whole thing led to the atom bomb. So Berkeley, Lawrence Berkeley Lab, was founded by Lawrence. He was a faculty of physics at Berkeley, and I have to do a digression and say that every Nobel Prize in Berkeley campus in science until this year was done at Lawrence Berkeley Lab. All the work was done in Lawrence Berkeley Lab, and these were people who had their labs up there. And there were about 10 of them, or 11 of them. And this year, Randy Sheckman won the Nobel Prize, and he did it on Berkeley campus. And I'm delighted that he got it. He's one of my favorite people on Berkeley campus. So it then became, when they separated themselves from Livermore, Livermore was extension of Berkeley, but it called itself Lawrence Livermore, and Lawrence and his wife were not happy about this, and Berkeley professors were upset to have military research above Berkeley, and in fact Berkeley was never like that, it was just an intellectual pursuit, and so half of the faculty up at Berkeley, half of the employees of Lawrence Berkeley Lab are faculty at Berkeley, and some of us have, either formal or informal faculty appointments in order to get the student stuff. But it is a campus of University of California. So I'm not a federal employee. I am a Berkeley, UC Berkeley employee. And I was also the first director of Cell and Molecular Biology Division at Berkeley Lab, and then now we call it also Berkeley Lab. And also became the associate laboratory director, and then all of biology went under me, and including the sequencing of the human genome, which, by the way, was started at DOE and not at NIH. It was their idea, and we were one of the first labs, and I was the director when they were doing it. And we still have a human genome center that, when I was director, I spun it off as a separate division because my division was getting too big, and I felt that I wasn't doing sequencing, and I needed to have somebody who was the director of that. So, and it's still going very strong. And anyway, I have stayed there for different reasons. And probably the first reason is that when I came to Berkeley, and after 2 years of postdoc, my husband, it was the Vietnam War. And my husband was in a thing called a Berry Plan. which allowed the doctors to enlist, but have to finish their research and whatever. And so I couldn't move. But also because I was doing such crazy stuff, like glucose metabolism, in the 70s, and nobody had any idea why I was doing it. What is she doing? Everybody thought it was the most boring stuff. And everybody was doing oncogene and things. So I'm not sure that many people wanted. I got one job offer, but I couldn't go. And so I went and interviewed for this job with Melvin Calvin, who had won the Nobel Prize for path of carbon in photosynthesis, and he had his own little building in Berkeley campus. He was a professor of chemistry. But he used to, every year he would get interested in something else. So he was interested in memory, in evolution, in photosynthesis,
So
and he got interested in cancer. Well, he would do them all at once, and then he would get people, and he would, So I went there to help them out to set up a tissue culture and do, so I went there as a staff scientist. And it was a very interesting time. I was pregnant with my second child, and he took a look at me and fired me. And I'm not going to talk about this because it's a long story. In those days, you could do this. He didn't believe women who were pregnant had any. In those days, we still didn't have any rules. And his own wife had had a miscarriage when she was a photographer, professional photographer, and she was in the field when she was pregnant. And she had a miscarriage, and they all blamed it on her being in the field. And he basically would not let me go up to the lab, and it was a very interesting story. But the good news is that he and I became the best friend he ever had. And he became one of my biggest supporters. And when they gave him $1 million to build a tissue culture laboratory, he basically gave it to me. And I helped build a lab that was up the hill, and it was mine. It was wonderful.
So do you remember what was your first grant on?
So the very first grant was when I was still in the round building with Melvin Calvin on campus. We were not allowed in those days because the national labs also used to have money, and they still do in physics, engineering, etc., but they never had it in biology, from government, from Department of Energy. But in biology, you didn't have the money. You had to go and get the money. Even from DOE, you had to apply. They didn't give any base money. But they had a very interesting policy that now people are trying to imitate it. And that they gave the money to the person and not because of what you wrote. When they thought somebody was smart and energetic and had some vision it was sort of like DARPA, And they had given money to Calvin to do the, and the entire use of radioactivity in medicine started from Calvin's work, because the Lawrence's brother was an MD, and we made radioactive material, fast acting, and that's how the whole thing started for the entire world. It was at Lawrence Berkeley Lab. They had really high-speed centrifuges, so they discovered LDL and HDL. We had a big group on lipoproteins, and it was discovered at Lawrence Berkeley Lab. People, and Calvin did what he did because he was associated with Lawrence Berkeley Lab. So it was a good thing for me to be there, and I did a lot of things in those days that I probably would have not been able to do in a university, and I stayed still because this concept that I have developed and has taken 35, 40 years was so alien to so many people that if I were in a regular university, I would have been given hell.
Do you recall the title of your grant, of the first grant?
It was something about what is the definition of normal. And and it went to NSF. And we weren't allowed to go to
This
NSF.
is the National Science Foundation in the US.
Yes, and there were people there who had a vision, I sent a grant, which I thought was a very good grant, to NIH, and they thought I was nuts. And even on the same subject. And because I was saying that, even early days in the 70s, I was saying that these concepts of terminal differentiation, these concepts of luxury molecule and housekeeping function, these concepts of induction and constitutive gene expression, the ideas of cell autonomous, none of them made sense. And that I wrote a review that was in 1981, and is a single author review, and it was based on the kind of stuff that I sent to National Science Foundation, and I got it. And apparently I wasn't supposed to get it, but that was my first grant. Somebody had the vision. It was only for 2 years. The vision to get excited about this. And they said, my goodness, nobody's doing this. And they gave me the money, and I was ecstatic. And of course I competed for money from Department of Energy and got it. And then later on got a big grant that now is taken away, but which is too bad, because it was allowing me to answer some of these very important questions. So now you know more than you wanted to.
No. So Mina, who were your role models and your mentors, if any?
That's why I love my English professor so much, because I honestly think that of the people I saw, Luigi Guarini was my professor, and he was a perfectly good mentor, but it took a lot before he decided that I really know what I was talking about, because I was all alone in his lab. Everybody else was doing bacterial genetics of ribosomal ambiguity mutation and streptomycin resistance and heavy, whatever. I was the only one, and he had postdocs before me who had worked on that project. I liked him. I thought he was very good. But the kind of role model that I can only count them on fingers of my hand. I had a math teacher at 7th grade in Iran. And she was fantastic. And she was the kind of person who could motivate you. And when you did something good, she had the passion and the oomph and whatever. And I loved her. Then I had the professor of literature. You see, that's why I went with Bissell. And he, I liked him a lot. And
so what did he do that was appealing to you? He was also motivating?
Well, it was, high school and primary school was very easy for me. And in those days, I still had some part of a photographic memory that apparently I had as a kid, but then I fell in my head, and I always tell people the reason I did all this crazy stuff. I cracked my skull, and I completely became deaf on my left ear, and all my life I have had to deal with this. And I also, at least the neurologists tell me, because ever since I was 8, I could not remember proper names. I had no problem with what I learned in school, science, this, that. Proper names, I still can't do it. It's like 90-year-olds, so this area that the left frontal lobe where you put things. So, but I lost a lot of my photographic memory that apparently I had, and I remember even part of it because everybody used to always test me. Is she for real? Does she memorize it ahead of time? But I kept some of it. And so it came easy to me. So what was interesting to me were professors who had passion and who could recognize people who were good without discouraging people who weren't that good.
Absolutely.
And that was so important for me. And we had, for example, a psychology and logic professor in 11th and 12th grade. And she hated my, she just didn't like me. And the reason was people couldn't understand her. So after she would leave, I would just give her a lecture to the kids. And one day she came back and she saw me doing this, and she was furious, But on the other hand, when you had people who saw something interesting and they, rather than trying to suppress it or judge it or change it, they encouraged it and they said, this is good. And he liked my professor of literature. He loved poetry, and he wanted to ask people to go. For example, we had oral exam
Yes.
as well as written exam. You know, we had something like 20 different subjects every year, and oral exam and written. So in the oral exam, he once, I remember, asked people to go and memorize a poem. So I walked in his room and I memorized something from Hafiz, who is
Wow,
one of our greatest poets. And it says, "I and the denial of the wine, what a story." You know, something like that.
Yes.
I just said the first line and he cracked up and he said, "Go." He gave me the highest mark. You know, they had that kind of energy.
Absolutely.
And they were appreciative of kids who were different. They
Absolutely.
didn't try to change me, which was lovely.
So with
And
Iran, you have a lineage of Khayyam, Omar Khayyam, another great
and Omar
poet
Khayyam,
of all times.
well, Omar Khayyam doesn't begin to compare with Hafiz.
Okay.
And Hafiz is our greatest poet, and we have others like Molavi and you know, Sadi and others, but they're hard to translate.
Yes.
Omar Khayyam was easy, and somebody took his quadrant, but he
Yes.
was a good poet, but he doesn't begin to compare
And
with
he was
these.
not only poet, he was also a mathematician.
Yes, a whole lot of these people were like that. I mean,
Yes.
you know that because of your own background.
Yes.
Yeah.
So, Mina, yeah,
yeah.
please.
And my English professor at Bryn Mawr, they wouldn't let the foreign student take that course because you
Yeah.
had to read a book and then write an essay every weekend and hand it in on Monday. And I thank my luckiest God, you know, fortune or whatever to have taken that. I insisted. I said I will take math and chemistry, those are easy for me, but I didn't know very much English. And she was amazing. She had charisma, she had passion, and it has stayed with me. One of the best things I have done. So these people were my role models.
Okay.
Yeah.
Yes. So, Mina, back to research. Your first paper on cancer was in 1971. What did we know about cancer back then?
Well, we knew it was the discovery of Ras sarcomovirus, right?
Yes.
And people were dumping, they had chick embryos or 3T3 cells. They were doing cell culture, but they were all cell lines, or even if they were primaries, they were a gumish of embryos. But then we were dumping this virus, Ras sarcomovirus, on the dish, and we would say, this is normal, this is malignant, where the malignant had little bumps, as you remember, focus-forming units, right?
Yes.
That's what I went to do my postdoc in. And that's what my professor had said, and he got into New York Times or something in those days about the protease. And that's how I showed that that's leakage and not secretion of a specific.
So that was one. So your second paper now on cancer was on patterns of glucose metabolism in normal and virus-transformed chick cells in tissue culture. This was in 1972. Were you interested in cancer metabolism?
Well, that's a very interesting question. It again comes from my curiosity. When I came to Berkeley, the question was, what would I work on? Everybody was trying to isolate what is the gene or whatever that made Ras sarcomovirus or these other DNA viruses. You know, DNA viruses, RNA viruses were the rage in the 70s.
Yes.
And I was next door to, in fact, a guy who, for various personal reasons, never did it, but he really showed the oncogene and should have won the Nobel Prize for it. But this is Peter Duisberg. He then started fussing about AIDS and stuff, and people just, you know, excommunicated him. But the point is that everybody was just wanted to do this. I mean, and I was pregnant again, and well, not pregnant. I had already a daughter, and and then soon after that, So when I went to the Calvin laboratory, I decided, or no, actually, even before going to Calvin's laboratory, no one in Harry Rubin's lab was doing this. So I read the literature, and I said, why is it that Warburg was so famous? And he talked about aerobic glycolysis. He won the Nobel Prize. Now, 10 years later, the literature was a mess. Somebody would say he was right, somebody would say he's wrong, somebody would say normal cells make aerobic glycolysis, they make lactate, somebody... And it just occurred to me that this was an area where I could actually try and learn about what are these fads in science that come and go, and why is there so much controversy. So the paper that I published actually after this in Journal of National Cancer Institute, while I was still in Harry's lab, it took that much longer. But the minute I went to Melvin Calvin's lab, I published this other one. Came from a study that I actually clarified a lot of the stuff in the literature. And I said that if you control for everything, like amount of glucose that goes in and the lactate that comes out, and you use the ratio, and you do this under very controlled conditions where you don't let the tumor cells go to high density. You keep the density, the rate of growth, whatever constant, and you do these things, then Warburg was right in saying it's aerobic glycolysis. So when I got to Calvin's lab, and I got a staff position job, even that year where he fired me, of course other people came and said, you can't do this, you know. And he would say, okay, then she's not allowed to go up. She didn't tell us, I was 1 month pregnant. I wasn't going to announce that, hey guys, I'm going to have a baby, you know. So what I did was that I realized that no one had looked at actually pattern of glucose metabolites as a measure of functional differentiation, okay? So why not? Everybody was saying those things that I told you, housekeeping function. I mean, glucose was a housekeeping. It just didn't make any sense to me. And I always say to people that one of the reasons I did some of the things I did is not only because I am who I am curious, and I was raised to always ask questions. That was encouraged in my family, which is, in a lot of places. I have a colleague in Canada who, she was the smartest kid, asked questions. The parents nearly destroyed her life because they wanted her to shut up. Kids are to be seen and not heard. And, it was not the rule in my family, My uncles would bring me Grapes of Rat to read when I was in 6th grade. It was that kind of stuff. So, this is a really a morality tale about how I got into glucose metabolism. The other thing I noticed was that it was the same thing in my lab. I never got to go to Cold Spring Harbor where my professor was going every 2 minutes because they all were competing about this ribosomal ambiguity mutation. I found that fine, but I didn't like it because it got them to do things that I didn't like. They made claims, or they were watching somebody else doing something, and then they were doing it, they were running and then trying to, And I just thought people are not taking time to think.
Yes.
They're not taking time to actually understand the essence of a big problem they want to answer. So that's why I chose this. So then I published that paper, and then I took the technique Melvin Calvin had developed for path of carbon in photosynthesis. And I also did something else which I probably know nothing about, but now I show it in my talks, and I will tell you later why that is. This is going to go on and on, Momo. How much do you want me to talk?
It's okay, you can.
Yes?
Yeah.
Right. So what happened was I had talked to the engineers, and I couldn't believe the way we do cell biology.
Yes.
And we opened the incubator door, the pH changes, the temperature changes, and I knew as a bacteriologist that 0.01 change in pH made all the difference in a different pathway or something, for crying out loud. So we made a round thing that we called the steady state machine. And we kept the pH and temperature constant, and it had 30 places, and for 35 millimeter place, and it had 2 septum.
Yes.
And in one we would add radioactive glucose, and then we bring it around and take it without changing anything, so we could do kinetic studies. And then we took these radioactive samples from normal and malignant and scraped them and put them the way Melvin Calvin had done. But I did the first experiment with animal cells, and these were chick embryos. And I did would run it in a big paper chromatogram. It was really the first system biology, you know. We would run it in one direction, we'd come the next morning, turn it around and put phenol and whatever, and you know, from my old chemistry days, I still remember those things. And then we would put a large X-ray film, and then we would get the metabolites as a function of time. And when we did this, it was quite clear that a pattern of glucose metabolism was cell and tissue specific. So in the liver, you got one pattern. In the muscle, you got another pattern. In the fibroblasts, you got... So this question of tissue specificity and housekeeping function, to call glucose a housekeeping function, early on led me to most of the other things I did later, because, and I began to talk to people about it and saying, this is really important, you know, but the eyes were glazed because everybody wanted to do. It was the beginning of molecular biology. It was the beginning of people were saying, you know. Then I asked the question, okay, so Warburg was right in aerobic glycolysis, but is it in fact that the respiration is messed up? And we did another experiment, again under this condition, with a lot of tritium. And we looked at oxygen transfer pathways. And under controlled condition, they didn't need to change. So aerobic glycolysis was correct, but that the respiration had to be, not that respiration doesn't go off in tumors or whatever, but it didn't have to be to increase that. So I said, what is it? I even wrote a paper about a shape of the cells in 2D and then 3D, and then we did electron microscope thinking, could this, you know, we made the shapes the same. I in fact published a paper on the rate of uptake that was shape dependent, you know, with Judah Folkman having done something on shape for growth regulation in 1978, and I did it in 1970. So, you know, we were sort of contemporaries that way. And so I showed that that respiration was not enough, but what was it? It wasn't shape, it wasn't... So to make a long story short, it was the rate of uptake of glucose. Now sugar gets taken up by both facilitated transport and by diffusion. And so the tumor cells were picking up a heck of a lot of glucose. And I said, gee, maybe this is the cause. So I took the glucose away from tumor cells, the pattern became normal. I increased the glucose uptake in the normal cells by going from 5.5 millimolar to 25 millimolar, and the pattern became malignant. I was so excited. I said, guys, pay attention, this is important.
This is not housekeeping.
It's not only housekeeping, it is controlling the pattern of aerobic glycolysis. So if Warburg wants to, you know, if people wanted to believe that it has something to do with cancer, this could have some importance. And I published this as a single author paper in Journal of Physiology or something. So it took me 30 years to come back to it, 35 years. And we just have our paper in Journal of Clinical Investigation in January on human breast. When I was doing this stuff in chick embryo fibroblast, even with all my energy and passion, I would stand in front of people and they would glaze because, you know, chickens and glucose, nobody wanted to lose. So that's how I chose mammary gland. And my postdoc, Joanne Emmerman, who came, knew about mammary gland to be my postdoc, and that's how we, little by little, weaned out. And I send all my incubators and stuff with one of my postdoc, who then is the head of a department at Irvine at UC, Manuela Green, and started getting interested in mammary gland because it's such a exciting tissue. And I began to ask the question of whether or not people were correct to say that everything was self-autonomous, etc.,
Yes,
etc.
so we will come back to this. So, but after these initial papers, you published numerous studies, including several nature and science papers. One of the nature papers was on the role of SARC in growth regulation. So we talked about it. This was in 1980.
84.
84?
Yeah. I did one in 1979, which wasn't necessarily a good paper, but it was in PNAS, and I said that viral carcinogenesis was multi-step, just like chemical carcinogenesis. But the first paper on the showing that the virus was not sufficient was in 1984 in
Yes.
Nature.
Yeah, that was also a second Nature paper. Absolutely, this one.
Yeah.
Inability of Rous sarcoma
To do-
virus to cause sarcoma in the
Yeah.
avian embryo.
Mhm.
Is this the one of your favorite papers?
It is.
Because
It
I
is.
know you talk
the paper
about-
you're talking about growth was
The first
in Nature,
one is in Nature paper.
and had to do with insulin, and it was a very interesting finding. In fact, we were probably the first who said tubulin has to be involved in growth regulation, and it was just a very chance discovery. I had a postdoc who was doing flow cytometry, and she was using the inhibitor that stops cells in mitosis and also affects tubulin.
Uh-huh.
And so she came to my room and said, it didn't work. It just doesn't make any sense. I gave the insulin, whatever, and we looked at it, and she dumped it in the wastebasket. And I went home, and I'm having dinner with my husband, and all of a sudden some light went on. And I said, oh my God. And I ran back to the lab to pull it out of the wastebasket before they cleaned it. And I said, hey, this means that tubulin is involved in growth regulation. And I wrote this little paper for Nature, and the editor said, well, it's interesting, and I may publish it, but you have to take your model away. And I wrote back, it was a very coarse drawing of a model. And he said, you're in 1977 or something like that. So he said, take it, eliminate it. I said, well, give me another model. And he published it. Yeah, so that was my first paper. And then
so that was the 1984 paper then?
No,
that was 1980. That's 1980.
This was 1977 or something. And then the paper I
The
told
one,
you about,
yes.
where I showed that
Yes.
hydrogen transfer pathways were not involved, I'm the first author, and it was in Science,
Okay, yeah.
in 1976.
Mm-hmm. But
Then
let's-
in 80s,
Yes.
there were these two papers
Okay.
that I cite all the time, and I still show those as slides. Because, Momo, you know that even now, there are people who believe there's only a single oncogene is enough.
Yes.
And I don't know, they had put the wool over their eyes. I have no idea why people should do that. These are some very famous scientists. They're very much that's why a lot of the stuff I talk about, and I have done 35 years of work, is not in the textbooks. Because the people who write the textbooks have a vested interest in glorifying what they themselves have done. And I don't have time to write the textbooks. So it's, that one showed that if I injected the virus with David Dahlberg, who was my postdoc, who then later went to law school. But he became a technology transfer guy because he had to be helped to do this stuff, but was a wonderful guy. So we did that paper. We injected the virus in the chick embryo, and we showed that it had PP60 SARC activity and everything else, but it didn't form tumors. And in those days, Nature and Science would take chances. It wasn't this thing that you had to have a famous person write and you had to kill yourself.
And to have 50 supplementary figures.
And to have 50 supplementary figures. They look at it and they say, this is an interesting thing. And in fact, when I published the next paper, which had to do with wounding and co-carcinogenesis, and went to Science, Koshland at that time was the editor, and they initially rejected it. And I wrote back and I said, show this to Bob Tejan or show it to one of your stars. This is a really important paper. It says that even in the chicken, something else is necessary, and wounding is a co-carcinogen. And it was one of the very first papers that actually showed the importance of inflammation and wounding and fibrosis and whatever else in tumor formation. And it was a really cute experiment. You know, all you needed to have was a chicken and a syringe and a brass sarcomavirus. And we showed that, you know, it comes here, and then when you wound, it comes here. And then we showed that the active component was TGF-beta. And that was also in Science in 1990. And it was the first paper we showed that TGF-beta has two functions.
Yes.
And people almost killed me for that too, because they were going to give it to patients to cure cancer. Because, you know, TGF-beta in normal cells they stop growth. But so that was the first. You're hearing my whole lectures.
That's fantastic.
That's good.
So while going through your papers, I noticed that you published with Dan Medina in 1987 on casein gene expression in mouse mammary epithelial cell lines, dependence upon extracellular matrix and cell type. How did you meet Dan?
Well, in the mammary gland conferences, right? Wow. Or in giving lectures, because by then I had started publishing quite a bit on glucose metabolism in mammary gland. When Joanne Emmerman came, we did what we did for chicken, we did it for mammary cells. And we published quite a few metabolic stuff and other things on mammary gland biology, and they were all very exciting. And then we started doing this floating collagen gel stuff that Dorothy Pitelka and Joanne Emmerman did the first on mammary gland, but before that, it was done by a guy called liver guy. He's in Pittsburgh, and the name will come to me, but the references are in my papers. He had done it for liver and had gotten some activity of hepatocyte. And so when Joanne did it first in vitro or something, she did a beautiful EM, and she could show that there was a little milk, and that the, you know, but nobody knew what was that due for. What was it?
Absolutely.
Okay. So we began to do studies where we took the cells, and I still show the pictures of Joanne from 25 years ago, where you look at one mammary cell that is making milk. It's gorgeous, it's polar, absolutely. It's secreting. It's
I love this slide.
Yeah, and then you put it down.
Yeah, then
it completely flattens. It forgets to make milk and whatever. So I began to ask, what is it that is missing? And I am really indebted to two of my postdocs. One was a cheek tendon guy who is still doing some work in my lab. And he was, you know, for the past 40 years. And his name was Rick Schwartz, and he had done all his studies in collagen, in tendon cells. And he came to be my postdoc, and we did some work together. We published a lot of stuff on collagen. And he published recently, I think, also. He published recently.
Yes.
And it's something very important.
Yes.
Is to spend all his career on this one topic. I love this man. So when he couldn't get funding, I brought him back to my lab and gave him a lab. And I became a little funding agency by letting him use what he wanted to.
Yeah, Anna, your former PhD student who is now in my lab, told me that he
Who is that?
Anna.
Anna, yes.
So she told me that he is very thoughtful person.
He is a lovely person,
yes,
and he is just fantastic. So he came back, but of course he didn't believe I understood anything. That was the cute part of it. When he was leaving my lab, he got an NIH grant to go to Jackson Lab. He said, "No, Mina, I'm leaving," and you know, and I said, "Rick, trust me. Just give me a little credit for running a lab." And then he came back in no time at all. He is thoughtful, he is very nice, and he can give you advice. He's a good biochemist. So that's how you know his paper for
Absolutely.
Anna. I didn't know Anna is in your lab.
Yes,
I'm so glad. She is fantastic.
She is, absolutely.
And we'll talk at lunch about Anna.
Yes, absolutely. So the same year when you published paper with Dan Medina, you also published your first paper with Zina Werb.
Yes?
Then
No, it was before that. With Zina, I published earlier.
1987, that's what I found.
Is that right?
Yes.
Yeah? Okay.
So then followed...
With Rabia Talhouk.
Exactly, who is professor at AUB.
Exactly.
Absolutely.
He's one of my most favorite postdocs.
So then followed several important papers published together with Zina. How did you meet Zina, and can you tell us about this very fruitful collaboration?
Yeah, so here is, so I was the first person to suggest. A lot of people were doing a lot of chemistry and biochemistry on extracellular matrix material. So I interrupted myself when I was talking about Joanne. So when Joanne came to my lab and did those experiments, and I began to say, what on earth is going on here? What is missing in culture? It wasn't only Rick Schwartz who taught me about collagen. It was another guy called Glenn Hall, who then later started working with killer African bees. And he's a professor in Florida and very, but he knew all about basement membrane. He was a developmental biologist. And he wanted to come to my lab to actually, because he had heard me talk about whatever it was, and he had gone to Berkeley, and so he taught me about basement membrane. So we began to think that maybe what's missing is the basement membrane. And maybe, this is what Joanne had not done, is even though the floating collagen gel was allowing this, why was it allowing this? So I began to do things with extracellular matrix, and I began to do things with, so we have a 1997 paper in PNAS where we 1987 87 paper in PNAS where we took different component of extracellular matrix and then later on did a dot plot and blah blah.
So back
blah.
then, where did you take that from?
Matrigel.
Oh, Matrigel.
Well, it wasn't Matrigel. It was called EHS tumor. Okay, and it was made in the mouse, and people in.
So it's in the mouse, not the rat.
It was in the mouse. Okay. It's a mouse tumor, and it makes gobs of extracellular matrix. And people at NCI, the names don't come to me. I haven't had enough sleep, but they had done the structure of this, and they had said it was similar to basement membrane. Yes. And you can't isolate basement membrane because by the time you do it, it's all cross-linked, and because there's so little around things, and it loses its ability to do the right thing. So I got these tumors and put them in the mouse, and we used to make the EHS tumor material ourselves, and we should have absolutely patented because of its functional stuff, okay? And especially when I did that 1992 paper with Ole Peterson that we will end up talking to, is we tried to patent it, the assay. And we said that it is such an important assay because you could distinguish between normal and malignant. And unfortunately, they didn't think it was important. And I could have been so rich, not in the lab, I mean.